Welcome, welcome, welcome. Oops, I'm sorry, I accidentally hit the button cutting off my stream again today. Sorry, I am back. How are you doing, folks? Good to see you. Good to see you. Sorry about that. I keep having these accidental things where I click buttons off. It's good to see you, folks. Everybody ready for class today? Congratulations on the first exam. I think the course as a whole did a pretty good job. And I was very proud of you this weekend. The grades are looking good. We've got a ton of A's and a ton of B's in this class, and very few failing grades. So everybody's doing great. Good morning, April. Good to see you. Hooter, good to see you as well. Jennifer, good to see you. Shavoni, thanks for coming today. It's great to see you. Fantastic. It is so good to see everybody this morning. Okay. So you can hear my sound well enough, right? Can everybody hear me pretty well? Let's see, what camera am I? Yeah, I'm using my microphone, my good microphone, so you should be able to hear me. All right. Welcome, welcome, welcome. So today we are talking about Chapter 5, Sensation and Perception. So we're going to continue on with our biological theme. And we're going to talk a little bit about the physiology of the senses. If you remember, I told you at the beginning of the class, experimental psychologists are the psychologists who study consciousness and sensation and perception. And so we're going to be talking about experimental psychology today. Good morning, Ryan. Good to see you. So what we're going to do is talk about the basics of sensation and perception of the basics of sensation and perception. We're going to talk about the location of the sensory cortices, which you should already know. And then we'll talk a little bit about the visual pathway, color vision, and the difference between sensation and perception today. Okay, let's go ahead and get started. If you'll remember from the first week of class, I suggested to you that Gustav Fechner was the first person to demonstrate that the mind was systematic. He was studying sensory processes. So he is not only the founder of scientific psychology, but he is the founder of experimental psychology. And what he noticed was this thing called a just noticeable difference, was the least amount of change in a sensory experience that could be detected half of the time. Now, it turns out your ability to perceive absolute thresholds, the ability to perceive anything of a stimulus, depends upon your situation. So your ability to taste salt in a glass of water will depend on whether or not you've eaten a banana or a piece of pizza. If you've ever turned off the lights at night, you'll notice how when you turn off the lights at night, the room gets completely dark. And then after a couple of minutes, you start to see shadows in the room. Your absolute threshold for perceiving sensory experiences changes. But what Gustav Fechner found was that your ability to perceive change in senses never, ever, ever changes. And his area would eventually become known as the field of experimental psychology. And if you look right underneath me, you'll see a little graph measuring absolute threshold. Now we're going to talk about 5 main senses in this class: your sense of vision, your sense of hearing, your sense of smell, your sense of taste, and your sense of touch. But you know what? There are also other senses that we talk about. We talk about a sense of balance. We differentiate thermoreception from these other senses, the sense of heat. And we also talk about nociception, which is the sensation of pain. So you can think of all of these as ways in which your body gathers information about the world, what's going on visually, what's going on orally, what's going on with smell and taste, what you can feel in terms of pain and balance. All of these are sensory, is our brain's way of measuring what's going on in the outside world. The purpose of our senses is to provide information about the environment that can be integrated, evaluated, and used to organize and direct our behavior. So we take in sensory information and then we act on that sensory information to think, to move, and to feel. Okay, now here's what I really want you to get from this lecture: the difference between what we call sensation and perception. Your ability to use your senses requires two basic processes. The first is called sensation, and the second is called perception. Sensation is the process of recording sensory stimulation into patterns of neural impulses. Okay? So, if you think about it, when you're seeing things, light is coming in your eyes. The light getting turned from light energy into patterns of neural impulses, of action potentials, is sensation. When you eat something tasty, chemical molecules will settle on your tongue and in your nose, and your brain will turn those chemical molecules from physical energy into patterns of action potentials, neural impulses that are then recorded in your brain. That's called sensation. Okay, and that's done by specialized neural cells called sensory receptors. Okay, so you have rods and cones in your eyes that turn light energy into wavelength energy into action potentials, right? You have hair cells in your ears that bend when sound waves move them, and they turn sound into patterns of action potentials. You have these taste receptors inside these taste buds on your tongue that respond to chemical information. You have olfactory receptors in your nose that also respond to chemical information. And then we have neurons in our skin. We have neurons that turn some information into the feeling of pain. We have some neurons that turn pressure information into, or sensory information on the skin into pressure. And we even have neurons in our brain that turn heat differences in our skin into the sense of temperature around our body. So this is what's known as this sensation process. However, that's not the final step in the process of understanding our world. You now have to take those sensory experiences and make meaning from them, okay? So you see a person wearing a black hat, a burnt orange shirt with two eyes, two noses, and a mouth, and you make the judgment that you are looking at Dr. Roddenberry on this screen. Your judgment of what it is you are looking at is known as perception. And so our ability to perceive the world is two steps. The first is turning the world into patterns of neural impulses, which are recorded in our brain. And then perception comes from inside out, and it's your brain deciding what it is you see, hear, touch, or smell. Does that make sense? Does that make sense? So, and when we talk about visual illusions or sensory illusions, typically what's happening is our brain is misperceiving or creating incorrect perceptions of what sensory experiences we are seeing. All right, so visual illusions occur at the level of perception. Okay, so from sensory receptor to cortex. So let's think about this. Sensation is the activation of sensory neurons that occurs at the level of the stimulus in the peripheral nervous system. So the neurons in my skin, when you activate them, okay, that's sensation, and that's occurring in my peripheral nervous system. And perception is the integration of this sensory information into meaningful patterns. And the cells that we talked about that turn this chemical, physical information into patterns of neural activity are called sensory receptor cells. Now, the process of turning these this physical energy into patterns of neural activity is known as transduction. So whenever you touch your arm and you turn that into a pattern of neural impulses, your sensory neurons are transducing that mechanical energy into a sensory experience. When light comes in your eyes, your rod and cone cells transduce that light energy into patterns of neural activity. We call that transduction. So I want you to know the difference between sensation, perception, and I want you to know what transduction is. Okay? Lots of different sensory types in your brain. So we have these things called mechanoreceptors. Mechanoreceptors turn physical energy into patterns of transduce physical energy into patterns of neural activity. So hearing, when wave information bends your hair cells, that's physical pressure energy moving these hair cells in your ear. When you are touched, body position, stretching of your arms, this is all perceived by these receptors we'll call mechanoreceptors. Now, the receptor cells that do taste and smell transduce chemical molecules into patterns of electrical activity. Those are called chemoreceptors. Now, we also differentiate visual receptors, which turn electromagnetic energy into patterns of neural activities. You know what? You even have sensory neurons located all over your body that measure your body temperature, and they give you the experience of warmth. These are called thermoreceptors. And when your external temperature becomes too high compared to your core body temperature, your brain gives you the experience of being hot. When your surface temperature is too low compared to your core temperature, your body gives you the perception of being cold. And you know what? We even have receptors located all along our body, which measure when the integrity of our body has been violated, when a knife or something comes through your arm or leg, or when something pierces your body. And these receptors are called nociceptors, and they're responsible for perceiving pain. So all of these receptor cells, mechanoreceptors, chemoreceptors, visual receptors, and thermoreceptors, all they do is they turn external patterns, external energy into patterns of neural activity through a process we call transduction. And all of this sensory information, with the exception of smell, is routed through this structure in your brain called the thalamus that we introduced in chapter 2. So all of these sensory experiences come through this part of your brain, in the middle part of your brain called the thalamus. All right? And then all of this information that's captured through these sensory types is then recorded and perceived by the different sensory cortices in your brain. Now, we showed this picture before, but I'm going to hit it again on this exam because it is important. So I'm going to have it, I'm going to reference this on the second exam too. I want you to know where all of the primary sensory cortices are in your brain. Now, when I say primary sensory cortices, this is where the sensory information comes from the body part specifically to the brain to begin its processing journey. Now, you have association cortices, which put these sensory experiences together, which allow you to come to a complete understanding of what you're looking at. But the beginning of the sensory process occurs in the primary sensory cortices. Okay? Now, I want you to know that we have a somatosensory cortex let's see, let's go the somatosensory cortex. If you look right here, it's the dark blue stripe. It's in your parietal lobe. This is where you sense all of the things that touch your body. When you're itchy, when you're in pain, when you're being tickled, all of this information is processed in your primary sensory cortex, located in the parietal lobe. Your gustatory cortex and your olfactory cortex, taste and smell, are actually processed in your frontal lobes. Your gustatory cortex here is right at the back of your frontal lobe, and your olfactory cortex on smell is right here in the front underneath your orbitofrontal cortex. Okay, now your auditory cortex is located in your temporal lobe. Okay, and your vision is processed in the visual cortex located in your occipital lobe. Now, I'm going to add one sense that we typically don't think about when we think about the five senses, but I'm going to talk about balance. Your ability to know when you're sitting upright or when you're sitting sideways. If you've ever been on a ride at the fair which spins you around and around and makes you dizzy, you have lost your sense of balance temporarily. And your sense of balance, your sense of where your body is in the world, is controlled by your cerebellum located beneath your cerebral cortex. So these are all of the sensory cortices. You'll notice I have five primary sensory cortices. We typically don't think of cerebellum or the sense of balance as one of the five basic senses that we all have, but it's a sense nonetheless. Okay, so I've got that down here as the sixth sensory cortex. Does everybody understand that? So here's the deal. We have these receptor cells, and these receptor cells transduce physical energy into patterns of neural activity. This process is called transduction. And some of these sensory cortexes turn physical energy into patterns of neural activity. They're called mechanoreceptors. Some of these sensory receptors turn chemical energy into patterns of neural activity. Those are called chemoreceptors. You have specialized receptor cells that turn electromagnetic information into patterns of neural activity. Those are called visual receptors. And then you have receptors that turn warmth and some sensory information into pain. And those are called thermoreceptors and nociceptors. All right, and all of these receptor cells then transmit that information to primary sensory cortexes, where these patterns of neural activity are processed. I hope that makes sense. Now, let's talk specifically about vision. We're going to talk about vision for the rest of the class, okay? On Thursday, we'll talk about the other senses, but today we're going to talk about vision. If you look, there's a picture of an eye beneath me. Okay, now your eye is a sensory structure. It's actually considered part of your central nervous system. And so on the outside of this eye, you have some fibrous protective covering of the eye, which is called the sclera. And then you have a clear part in the front called the cornea. And then you have a structure in the back, a hard structure in the back called the choroid. And that's contains the blood supply for the eye. Now, information comes in through this clear covering called the cornea through a hole called the pupil in the eye. And the pupil in the eye, if you'll look at my eye, you'll see this black dot in the middle of my eye. That black dot is actually a hole leading into my eye that allows light information to go from the outside of my eye to the inside of my eye. If you look around that black structure, I have these nice blue eyes. My blue eyes are composed by my iris. Each and every one of you has a colored band of tissue around the pupil. This is called your iris. Now, your iris can relax or get tighter to increase the size of this hole, this pupil, or decrease the size of this hole to allow the proper amount of light into your eyeball. And so the first thing that happens is our eyeball controls the amount of light energy that gets into our eyes. If you've ever walked outside of a movie theater on a Saturday afternoon after watching a matinee movie, and as soon as you walk out of the movie theater, the world seems so incredibly bright for a second and it's hard to see. That's because your pupils have been large sitting in that dark movie theater, and as soon as you walk outside, your eyes are now receiving too much light energy. So what happens is your pupils, your irises constrict and make that pupil hole just a little bit smaller to decrease the amount of light energy that gets into your eye. Now, this light information comes through this pupil, is focused by this lens onto the very back part of your eyeball. Think of the back part of your eyeball as a movie projector screen. And what we want is we want the image, the movie image, to be projected perfectly and clearly on the back of the eyeball. The lens, its job is to focus that light energy perfectly on the back side of your eyeball. Now, if any of you are like me and you have glasses or what we call corrective lenses, what happens is those corrective lenses assist your lens, which doesn't focus the light energy perfectly on the back side of the eyeball. So if you have vision problems, if your eyes are 20/40, 20/60, 20/80, 2100, 2200 like my eyes, and you need corrective lenses, it's because the shape of your eyeball is such that your lens doesn't focus that image of what you're looking at perfectly on the back of your eyeball. And so you need a second lens to assist the first lens. Now, you actually have this structure, this little muscle structure on the outside of your lens called your ciliary muscle. And what your ciliary muscle does is it bends the lens so that when you come really close up to objects, you can focus and see them very clearly. If any of you are old like me in your 50s, or if you have talked to your parents, you'll notice that they probably have trouble reading things up close and they need to use bifocals. The reason that people need to use bifocals as they get older is because their lens that focuses light on their eye becomes stiff, just like every other joint and muscle in your body. Your lens becomes stiff as you get older. And so your ciliary muscles aren't able to bend that lens to focus the image right on your eyeball. So everybody with normal vision, or actually everybody with normal and abnormal vision, as you get older, you're going to need reading glasses. Because when you get up close to things, your lens is not going to be flexible enough to be bent by these ciliary muscles, and you're going to have corrective reading lenses. Okay, so this is what's in your eye. You've got this big structure called a sclera, and the front of it is a clear, hard structure called the cornea. Light comes in through this protective structure, comes in through that black hole called a pupil, and is focused by the lens on the very back part of your eyeball called the retina. Okay, now the retina is neural tissue. We actually consider the retina to be part of the central nervous system. And the retina is neural tissue on the back of your eyeball containing visual receptor cells called rods and cones. Okay, now if you are looking right at me right now, if you are looking at the screen and you see me and you are looking directly at me, I am focused on a very specific part of your retina called the fovea. And the fovea is the part of your retina where images fall when you are looking straight at them. Now, the fovea doesn't have support cells or blood vessels in this area. It just has tons and tons of rods and cones. And because it doesn't have all this extra stuff there, like the blood vessels and the support cells, it has even more rods and cones. And this is why when you're looking straight at something, it seems so clear and detailed. And if you look at things in the peripheral part of your vision, they're going to be a little bit blurry. And that's because they're falling on part of your retina that doesn't have as many rods and cone cells. Now, if you look at this picture right underneath me, what you're going to notice is that sort of in the middle at the bottom, there's this thing called an optic nerve. Do you see the optic nerve? It's listed on the left side of the picture. The left side of the picture, there's optic nerve. And the optic nerve is where the rods and cones send their information out of the eye back to the visual cortex where you can perceive vision. Now, it turns out that this place in your eye where the neurons exit your eyeball doesn't have any rods or cone cells. And so this is sort of a blind spot in your field of vision. So if you close one of your eyes, the other eye looking out, there's actually a spot in your field of vision where you don't have any rods or cones. And in fact, it's going to be to the outside of your head. So if I have my right eye open, it's going to be on my right side, kind of out here is a blind spot where I don't have any rods and cones. And if I close my other eye, and so now you've got my left eye open, there's a spot to the outside of my body, right here about 15 degrees, where there is also no rods and cones perceiving the light information coming into my eyes. So we actually have these two blind spots in our eye. Okay? Now let's talk a little bit about these rods and cones. These rods and cone cells are very specialized neurons that have proteins in them. And these proteins are light sensitive. And so when these proteins are energized by light information, by photons, if you will, which is packets of light information, photons cause these proteins to generate action potentials. So you have these proteins in your rods and cones called opsins, O-P-S-I-N-S. And these opsins, when they are stimulated by light photons, they generate action potentials. And so that's how light energy is translated into patterns of neural activity. Every time light comes into your eye and hits one of these rod or cone cells, it energizes the protein in that eye cell, and that protein generates an action potential. Okay, now we have two kinds of visual receptor cells in our retina. And what you'll see below me is a picture of a retina. Now, rod cells and cone cells. Now, your rod cells are more numerous outside the fovea. So when you're looking at something, when you're looking straight at something, that image is falling mostly on cone cells. Things outside the center of your vision, your peripheral vision, those images fall on a part of your retina that contains cone cells. So your rod cells are located mostly outside the fovea, and they work very well, very well in low light. Very low light. And these rod cells contain this protein called rhodopsin. And rhodopsin is very sensitive to photons. In fact, they say that one photon can energize a rod cell and cause it to send an action potential. That's how sensitive these cells are. Now, located inside your fovea, when you're looking at something, are cone cells. And you'll see that they're shaped kind of like cones as opposed to rods, looking at the picture over here. And they're located in your fovea. Now, the proteins, the opsins located in the cone cells aren't nearly as sensitive to photons of light energy as are rod cells. So they need more light to generate action potentials. But they have three different types of opsins. So you actually have cone cells with three different kinds of opsins. And these opsins each respond to light of a different wavelength. And these three different opsins are what create your perception of color vision. Okay? Now, have any of you ever seen an after image? Have you ever, any of you ever done the experiment where you look at a green background for 30 seconds and then you turn away and you're supposed to look at a white background? Does anybody know what you see if you look at a green color for 30 seconds and then turn away and look at a white background? What are you going to see on that white background? Does anyone know? Does anyone know? Have any of you ever seen that video illusion where you look at a white and black sketch that doesn't make any sense, and when you look away, it turns into a picture of Jesus? I don't know if anybody's ever seen that visual thing. There are these things called after images. If you look at green for a long time and then look away at a white screen, you will actually see the opposite color to green. You will see red. All right. Now, here's the weird thing. We actually have these cells called ganglion cells in our retina, and their job is to turn off one visual receptor cell when they're receiving information from another receptor cell. And so when one receptor cell is seeing red, when your red cone cell is seeing red, your ganglion cell, not only is it sending the red information to your brain, but it actually turns off the receptor cell that sees green. It actually impairs that functioning. And so your ganglion cells actually work together to coordinate the activity of your rods and cone cells. And so a ganglion cell receives input from 1 to up to 50 visual receptor cells, depending upon the location. And what it does is when it receives information from some cells, it turns off the other cells so that they can't, it impedes the functioning of the other cells. And so what it does is it allows us to sort of manage the input from our eyeballs. Okay, so we have rods and cone cells that are sensitive to light, and then we have ganglion cells that coordinate the activity of these rods and cone cells. All right, now your rod cells contain rhodopsin, which is very, very sensitive to light photons. And so your rod cells do not need much light at all to allow you to see images. However, your cone cells, which give you the perception of color vision, contain 3 different types of opsins that are not as sensitive to light energy. And so they need more photons to be stimulated. But these 3 different types of opsins give you the perception of red, green, and blue. Okay, so let's talk a little bit about these cone cells. Now, your cone cells contain 3 different kinds of opsins: a short wavelength opsin, a medium wavelength opsin, and a long wavelength opsin. So if you'll see the picture that I've got over here, what you have is a picture of the visual light spectrum. All right. So electromagnetic energy, which is what light is composed of, comes in many different forms with different sizes of wavelengths. And wavelength is the distance that a light electromagnetic energy has from trough to trough or peak to peak. Now, it turns out that there's lots of electromagnetic energy in the world that you do not perceive. So, for example, you do not have infrared vision. All right, you do not have X-ray vision. So, X-rays, infrared rays, radio waves are all forms of electromagnetic energy that are bouncing around the world. However, your rods and cones, the cells in your eyeball, are sensitive to electromagnetic energy in a very narrow wavelength we'll call the visible light spectrum. So if you measure electromagnetic energy, what the visual light spectrum is, it's any type of electromagnetic energy that goes from 400 nanometers to 700 nanometers. Any electromagnetic energy that falls within that range activates these rods and cone cells, and so we call this the visible light spectrum. This is where you can see light. Now, it turns out that these cone cells, these three different kinds of cone cells, respond to light in different parts of this visible light spectrum. So real quick, let me step back. Your rhodopsin, which is in your rod cells, is sensitive to any type of light energy in this visible spectrum. However, your cone cells have three different types of light sensitivity. So some of your cone cells are what we call S cones or blue cones. And what they do is they respond to the shortest part, the part of the light in the part of this visible wave, the visible light spectrum, the short end of the light spectrum. And that gives us the perception of blue, if you will. We have M cones, and they are responsible for responding to light in the middle of the visible light spectrum. And so they give us the perception of green, if you will. And then we have these L cones, and they are most sensitive to long wavelengths of light. And this sensitivity results in the perception of red. So in a sense, you can think of yourself as having blue cones, green cones, and red cones. Now, if any of you are any of you colorblind, have any of you been born with color blindness? Color blindness. Now, people who are born with color blindness are usually red-green, have red-green color blindness. That's the most common kind of color blindness. And what that means is they don't have the two different kinds of options in the red and green cones such that they can distinguish the color red from the color green. So they are missing in their genes, their genetic structure doesn't create visual cones with the red or green option. And so they can't differentiate light in the red wavelength from light in the green wavelength. And so that's why some people are born being colorblind or having red-green color blindness. Okay, John, you have red-green color blindness. You know, the gene that creates red-green color blindness is located on the Y chromosome in the 23rd pair. And so guys are going to be more likely to demonstrate color blindness than are females. You and your friend had difficulty playing Uno because of how dark the green was on the card. Absolutely. You know, John, I play golf with a fellow who has red-green color blindness, and he uses colored golf balls so he can see them easier. And he can't tell the difference between a yellow-green golf ball and an orange golf ball. So we'll be out on the course sometimes. He'll be playing an orange golf ball, and he'll walk up on one of those yellow-green golf balls and think it's his because he can't differentiate between these two colors. And so that's what occurs, John. You don't have the proper options functioning in your red and green cone cells. Now, here's the deal. Let's talk about how this information travels to the back of your eyeball. Okay, now the information comes in through your eyeball and the photon information, and it registers the rods and cone cells on your retina. Now, each of these rods and cone cells then connects with a ganglion cells, and ganglion cells sometimes gather information from just one rod or cone cells in the fovea of your vision. And some of these ganglion cells actually capture information from up to 50 of your rod cells located in your peripheral vision. And so in a sense, these ganglion cells coordinate the functioning of up from 1 to up to 50 rod or cone cells. And they then send this information out of your eyeball through a structure that becomes the optic nerve. And so you'll see the arrow right here. You'll notice that the arrow coming down shows you the light. The orange arrow comes down and shows you the light hitting the retina. And then you see the little black line with the arrows, and that shows you how these structures then transmit the information up to the ganglion cells, and the ganglion cells connect together and become and create axons that leave the brain in what's called the optic nerve. Now you see where these axons are leaving the brain. We don't have any rod or cone cells. This is why we have a blind spot in each of our eyeballs. And this is where the optic nerve is leaving the eyeball. And so the optic nerve leaves the eyeball and heads back to the front part of the thalamus. Right in front of the thalamus, underneath, right by the hypothalamus, is a structure called the optic chiasm. Now here's what I want you to understand. Your left field of vision, what we call your left hemifield, is perceived by the right part of your brain. And your right field of vision, called your right hemifield, is perceived by the left half of your brain. Now, here's the deal. Each eyeball sees the left half of your world and the right half of your world, right? So my left eye sees the left half of my world and needs to send that information to the left side of my brain. The right half of my world needs to send that to the left side of my brain. And my left eyeball also sees both halves of my brains, and it needs to send the information from the left half of my visual world to my right brain, and the right half of my visual world to my left brain. You know what? The other eyeball also sees the left half of my world and the right half of my world. And if you look at this picture underneath here, what you'll notice looking at these little red and blue egg-shaped cells they show that the eye is looking at. So what you'll see is that each one of your cells sees the left half of your world and the right half of your world. Now, each of these neurons needs to transmit, each of these eyes needs to make sure that it routes the visual information to the correct hemisphere. This rerouting of each of your eyes occurs in the optic chiasm, where each eye switches and sends the neurons containing the information from the correct hemifield to the correct hemisphere of the brain. And this occurs in the optic chiasm. Now, the information then travels from the optic chiasm to your thalamus. Actually, a very specific part of your thalamus called the LGN, the lateral geniculate nucleus, which we're not really going to talk about in this class. But the information goes to your thalamus, where it's represented in a visual map. Okay, imagine taking a grid and marking out your visual world in terms of coordinates. So there's a map in your thalamus where you can record everything that occurs in the different coordinates of your visual field. And then that map synapses with a bunch of other neurons that then head back to your primary visual cortex, where you have another map with coordinates where you record everything that you've seen in every part of your visual world. This is called the primary visual cortex. And this is where you develop a map of all the things that you see in your visual cortex. Now, I want to stop and make one small note here. Not all of your neural fibers go from your eyeballs all the way back to your occipital cortex. If you'll remember when we were talking about chapter 3 a couple of weeks ago, sleeping and dreaming, some of these nerve fibers that come from your eye, from your eyeball, actually stop at your hypothalamus in a structure called your suprachiasmatic nucleus. Remember, SCN? And that light information is recorded by your suprachiasmatic nucleus to let you know when it's time to sleep and when it's time to wake up. Well, you know what? There are also some other fibers that stop in the bottom of your thalamus in a structure called your superior colliculus. This is a structure in your thalamus where your vision coordinates to the rest of your senses. So if you think about this, you've never thought about this, folks, but the things that you see in your visual world, in order to grab those things, you need to be able to direct your eyes and your hands to where the things are in your visual field. Okay, so in order for me to pick up my coffee cup, my eyes have to tell my hands where to go in space so that I can pick up that coffee cup. So I look at my coffee cup and my eyes direct my hands to the correct area in space so that I can get my coffee cup. Your eyes are connected to your other senses in this structure called the superior colliculus. If you don't have this, if this superior colliculus gets damaged, you will be able to see things, but you won't be able to grab them in your visual space because your eyes will not be connected to your other senses and your other motor behaviors. Okay, now when you hear a sound, your eyeballs know exactly where to look to see that sound. Again, this is controlled in the superior colliculus. Okay, does that make sense? So think about it. Your eyes, the information coming from your eyes is being processed by at least 3 different areas. The first area is your suprachiasmatic nucleus, which uses visual energy to tell you when it's time to wake up and go to sleep. The second part of your brain connects your eyes to your other senses so you can know where to look when you hear sounds, or so you can know where to move your hand so you can pick up an object and use it. And then the third place that your eyes send information is to your occipital cortex so you can have the conscious experience of seeing things. I see the computer screen. I see Dr. Roddenberry. That happens in your primary visual cortex. Now, the primary visual cortex just processes movement, location, orientation, and color. We call it your primary visual cortex because it begins the process of perceiving what it is you see. And as you look at Dr. Roddenberry right now on this computer screen, your primary visual cortex is processing the orangey, ugly color of my shirt. Your visual cortex is processing the lines in my face. Look at how my mouth is composed of lines that run this way, horizontal lines. Then I have diagonal lines that make my nose. And then I have circles that make my eyes. And then I'm moving all over on your screen. All of these are the primary characteristics of the visual object that you are looking at. And this is all processed in your primary visual cortex. Now, the weird thing is, you have just processed me as lines and shapes and colors and movements. But the visual process is not over. You now need to figure out what it is you are looking at. And that's where perception comes in. And what we do is we try to make sense of what it is we are looking at. Okay? Now, the weird thing is, your sensory experience starts from the outside and comes in, and you perceive things. And then your brain tries to make sense of what these sounds are or visual sensory experiences that you see are. If you'll remember when we watched that video a couple of weeks ago, remember when they said, I think Brexit is a really terrible idea. Do you remember the first time you heard that? You didn't know what you were listening to. But after hearing the fellow say that one time, you were then able to hear that metallic mechanical voice and understand what exactly it was trying to say. The second time, your brain was able to make guesses about what it was you were hearing. The same thing happens with all of your senses, and that same thing happens with your visual senses. And so there are different ways in which your brain tries to make guesses to understand what it is you are looking at. And so you can think about your visual system as having these sort of principles that cause us to make guesses about what it is we are looking at. Now, there are two basic groups of of perceptive processes that go on in your brain when you're trying to make sense of what it is you are seeing. So the information comes into your visual cortex. It's then transferred to your temporal lobe, which tells you what it is you're looking at. And then that information is transferred to your parietal lobe, which tells you where it is you are seeing that object. And all this information is put together, and you try to make guesses about what it is you are looking at. Now, there are 5 basic ways in which we organize our visual experience. And these have become known as the Gestalt principles of grouping. And they're listed right under here and described in your book. So we tend to group things based upon how close they are to one another. And so if you look over in A, you'll see instead of seeing 15 dots, what you probably see are 3 groupings of 5 dots. Your brain is already making guesses about what it's looking at. It's saying, I'm not seeing 15 blue dots. It says, I'm seeing 3 groups of dots together. Your brain also organizes things into similarities and differences. So if you look at B, you'll notice that you see 2 different designs. You don't see one design, you see 2 designs based upon similarity. In fact, to me, this looks like sort of a bizarro picture of the beach. And I see the red lines as the ocean and the yellow squiggly lines as the beach, if you will. Now, you'll notice in C, what we know, they've provided the little green line going behind the thing so you can see what they're talking about. But if you were to see this pink cylinder and this green bar poking out either side of it, you wouldn't say it was 2 green dots on either side of that pink cylinder. You instead say that you see a long green piece of tape going behind the cylinder. Okay? Now, if you look at the triangle shape, really what you're seeing are 3 arrows that are facing in different directions. But your brain puts in a shape. Your brain closes out this structure to see a triangle instead of 3 angles. And then if you look in the bottom picture right here, your brain sees an imaginary square, even though there isn't a square there. So if you think about it, looking at these pictures, there are really 15 blue dots, but your brain makes them into 3 groups of dots. There are just some squiggly lines and some straight lines, but your brain turns it into an ocean picture with the beach and the water. Your brain sees a pink structure and 2 green structures, but instead it makes 1 green structure going completely behind that pink structure. Your brain sees 3 red arrows, but turns those 3 red arrows into a triangle. And your brain sees 3 circles with cut out pieces facing one another, but your brain turns those structures into a square, an imaginary square shape that you see. In a sense, these are all examples of your brain trying to guess what it is these sensory shapes are looking at. So when you perceive things, your brain tries to make sense of the patterns of light and dark, red and blue, and the movement and non-movement that you see in your world. And so these Gestalt clues demonstrate how our brain makes perceptual guesses about what it's looking at. And we do the same thing when we heard I think Brexis is a really terrible idea. Your auditory cortex took that information in and sent it to your temporal lobe, which then tried to make a guess about what it was looking at. Now, we do the same thing with visual perception as well. Now, we have 2 kinds of binocular vision. The first is called binocular disparity, or what we would call true depth perception. Your left eye and your right eye are both seeing the same object, and they're sending slightly different pictures back to your brain. So, if you close your eyes and look at this screen and switch left eye to right eye as you're looking at Dr. Roddenberry, what you're going to notice is Dr. Roddenberry appears to jump a little bit. And that's because your left eye is seeing a slightly different image than your right eye. This is what we call binocular or retinal disparity. And what happens is your brain perceives the slight difference in these images and makes a guess as to how far the object is away from you. Now, we can actually, after a while of seeing images using binocular disparity, your brain gets good at figuring out the clues that also demonstrate distance. And so we have several monocular depth cues, that is, ways in which our brain guesses about how far objects are away from one another. And what you can do and what artists do is they use these organizing principles to create the illusion of depth where it doesn't exist. And what I've got listed over here are the monocular depth cues that your brain has learned over time to guess how far objects are away from you. And if you look right underneath me, there are descriptions of each of these 6 different visual cues that your brain can use. So, for example, okay, what I can do is I can create a right here. This is a landscape, and if you'll notice, I have 2 mountains here. Which mountain is closer, Mountain A or Mountain B? Well, you know, Mountain A is closer because it's occluding Mountain B. It's getting in the way of Mountain B, right? And so we would call that, we would call that, visual occlusion, right? All right, now height and field. I can go back and I can draw a fellow here and I can draw a fellow here. Which fellow is closer? This fellow is closer because he's lower in the field of vision. So you can use height to judge which is closer or farther away. Now we also know that these 2 fellows should be about the same size, right? And so we know if this guy is twice as big, this guy down here is twice as big as this guy, we know that he must be closer to us because he's taking up more space on our retina than this guy back here. And then you know what I can do? I can add a road in here. And you know roads have parallel lines. But if you look at a road going off into the distance, what you're going to notice is that the lines, the parallel lines, begin to run together. And that gives us the perception of distance. Now, we all know that this road, the lines are going to stay parallel. But as it recedes off into the distance, it looks like these 2 lines kind of get close to one another. And then we can use changes in pattern. So, you know, there are lines on a road, but as you get farther off, those lines are going to get closer together. And so I can use lots of tricks to make you think that this is a 3-dimensional picture. I can use parallel lines getting close to one another. I can use a pattern getting smaller. I can use the fact that one guy is bigger than the other or lower in the visual field than the other. I can use the fact that this mountain covers up this mountain. And all of these are clues in which your brain uses a prior experience to make guesses about how far objects are away from you or close to you. I hope that makes sense. All right, I am sorry I've gone over my time. I did not mean to go over my time. But I just wanted you, for this class, I want you to understand the organs, the neurons in your eye that are responsible for light energy. I want you to know the difference between rod cells and cone cells. I want you to know what ganglion cells do. And then I want you to know the pathway that vision travels as it goes from your eyeballs to your optic chiasm, to your thalamus, to your visual cortex. And I want you to know that vision doesn't just come from the outside, but we actually make guesses about what it is we see and hear and taste. And so we have a sensory process as well as a perceptive process. All right? Does that make sense? I feel like I sped over this and didn't do a great job. I hope you understood what I was talking about today, and I hope you learned a little bit from today's lecture. Does anybody have any questions? Don't forget we have webinar Wednesday night and Thursday night. I hope to see you at webinar on Wednesday night at 7 p.m. If you can't make it to webinar, I will see you in class on Thursday morning. Any questions? If not, I'm going to go ahead and say goodbye. Have a great day, and I look forward to seeing you tomorrow evening at 7 p.m. Take care.